Hey everyone, and welcome biology students to this explainer. Today, we're diving into a really fascinating study, doing a formal scientific review of how researchers are using in silico virtual screening to hunt down natural, plant-based antiviral compounds against the dengue virus. If you've ever found yourself wondering how computational biology is just totally accelerating the discovery of new medicines, literally before anyone even steps foot in a wet lab, well, you are definitely in the right place. Let's get into it.
To keep things structured today, we're going to follow an adapted IMRAD arc. First, we'll look at the dengue treatment gap, then filtering via Lipinski's rule and the principles of molecular docking. After that, we'll hit the medicinal plant docking results, predict protein binding stability, and finally, wrap up with translating all this to clinical treatments.
All right, moving right into part one, the dengue treatment gap.
So, 50 million. That number is just absolutely staggering. Every single year, there are roughly 50 million cases of dengue infection worldwide. Dengue is a flaviovirus, mostly transmitted by Aedes mosquitoes. And honestly, despite this massive global burden, there is still a complete unavailability of specific antiviral drugs or even highly effective universally applicable vaccines to treat or prevent it. Medical professionals are basically stuck relying on supportive care.
And that total lack of targeted therapeutics is a huge issue because of how this disease actually progresses. Lots of infections just cause mild, flu-like symptoms, sure, but they can rapidly and incredibly unpredictably escalate into severe conditions like dengue hemorrhagic fever, or DHF. We're talking severe bleeding, blood plasma leakage, which can eventually lead to shock and even death. Because of that, discovering accessible, effective antiviral treatments is just a massive, urgent priority for virologists right now.
Okay, on to Section 2, filtering via Lipinski's rule. To hunt down some novel therapies, the researchers behind this study actually turned to traditional Indian medicinal plants. But obviously, they first needed a way to figure out which of the hundreds of natural phytochemicals actually possess the chemical properties of a real functional drug. So using this free chemoinformatics software called Mall Inspiration, they ran 35 different compounds from plants like neem and papaya through what is known as Lipinski's Rule of 5. As biology students, you guys probably recognize this as a super vital preliminary filter. It basically evaluates pharmacokinetic parameters, things like molecular weight, hydrogen bonding capacity, lipophilicity, just to make sure the chemical structures can actually be absorbed by humans for drug development. Out of the 35 compounds they tested, 27 successfully passed this strict filter.
That brings us to Section 3, Principles of Molecular Docking.
For those 27 natural candidates that did show drug-like properties, the study moved to the next critical phase, which is high-throughput computational screening. Now, this molecular docking workflow, it is an absolute game-changer in modern pharmacology. Instead of spending decades stuck in a lab doing trial and error, they use software like Autodoc to virtually predict exactly how these plant-derived compounds or ligands will physically interact with specific target proteins on the dengue virus. Here, they targeted the viral envelope proteins, which the virus uses to enter our cells, and the methyltransferase enzyme, which it needs to replicate its RNA. By predicting these molecular interactions in silico first, this workflow saves just a massive amount of time, energy, and resources.
Now let's look at Section 4, Medicinal Plant Docking Results. You've got to see this data, because the sheer potency of the compounds derived from the neem plant, scientifically Azadirachta indica, is just remarkable. The top compound, meliosinan hydride, showed an incredibly low binding energy of negative 9.2 kilocalories per mole against the viral envelope protein. That compound, along with nimbocinil, literally blew the synthetic reference drug, acetaminophen, right out of the water. We're also seeing some really promising interactions from compounds derived from Andrographis paniculata and Tennisbora cardifolia. But honestly, the neem plant derivatives were the undisputed stars of the simulation here.
Just to contextualize those numbers for a second, remember that in pharmacology, a really low, meaning highly negative binding energy score, paired with a low inhibitory constant, or Ki, means the ligand binds incredibly tightly to its target. Thinking about this at a macro level, if a compound can bind tightly to that viral envelope protein, it might successfully block the virus from ever fusing with the host cell membrane. Or if it binds to the methyltransferase, it stops viral replication dead in its tracks. This is the exact mechanism of action virologists are constantly looking for in a viable antiviral drug.
Which leads perfectly into Section 5, Predicting Protein Binding Stability.
To really grasp the physical chemistry behind these awesome numbers, we have to visualize the spatial interactions.
If you look at the actual 3D docking snapshots generated by the researchers, you can clearly see compounds like maliocin anhydride and nimbacosinol just nestled so tightly within the active, highly specific hydrophobic pockets of the dengue viral proteins. I mean, they aren't just randomly bumping into the protein, they are fitting perfectly, literally like a key into a lock. And zooming in closely at the micro level of those 3D models, we can see the actual chemistry at work. Take maliocin anhydride, for example. It successfully forms 3 distinct hydrogen bonds with specific amino acids, arginine, isoleucine, and glycine, right inside the binding pocket of the viral envelope protein. And as we know, the presence of multiple hydrogen bonds highly predicts complex stability. Basically, it tells us that this compound won't just easily dislodge. It's going to hold on tight, physically and chemically preventing the virus from completing its structural mission of infecting a host cell.
Finally, Section 6, translating to clinical treatments.
So, the million-dollar question: what does all this actually mean for the future of medicine? How do we take these computational in silico findings and turn them into real, tangible clinical treatments for patients? Well, the potential here is huge. Today's pharmaceutical landscape relies super heavily on synthetic drugs, and those often come with astronomical development costs, much higher toxicity risks, and, maybe most critically, an increased susceptibility to viral drug resistance. But utilizing proven natural phytochemicals, like the ones highlighted in this study, offers a totally eco-friendly, highly cost-effective alternative. Because these natural structures evolved in biological systems, they tend to have much higher historical safety profiles. Plus, viruses usually struggle way more to develop resistance against complex, naturally derived compounds compared to synthetics. It's a win-win.
I'm going to leave you with this final thought today. Computational docking has clearly done its job here, and done it beautifully. It's sifted through all the noise and successfully pinpointed maliocin anhydride and nimbacosinol as highly potent natural candidates against the dengue virus. But at the end of the day, a computer simulation is just step one. The big question now is how quickly can the scientific and pharmacological communities take these exact findings, design the necessary in vitro lab experiments, and then launch the subsequent in vivo clinical trials to safely bring these antiviral treatments to the 50 million people who desperately need them every single year. The blueprints have been drawn, now it's time to actually build the treatment. Thanks so much for joining me in this explainer, and keep exploring the amazing world of computational biology.